Good morning, everyone. Welcome to the second day of the Advanced Photoelectric Sensor Integration and Bioimaging Newton Fund Researcher Links Workshop. I'll be chairing the session this morning. I'm Nigel Copner from the University of South Wales, and I lead the Optoelectronics Research Centre there. To kick off the session this morning, we've got a plenary session from Professor Jonathan Cooper, and he'll be talking about low-cost, mobile phone-enabled infectious disease diagnosis in remote, underserved communities. So it's ironic that he's actually calling in today virtually, so hopefully this should work quite well. If it doesn't bode well. Jonathan, over to you. Thank you very much.
Okay, thank you very much. Thank you. There was a slight echo
You're on mute, John.
that I could hear on your...
Jonathan, you're on mute.
Can you hear me now? Yes? Very good. I could hear a slight echo on you. Can you hear an echo on what I'm saying or not? No, you're on mute.
Sorry. There's no echo on our side, John.
No, okay, good. Okay, well, thank you. Can I say thank you to the organisers for inviting me, and a particular thank you for letting me do this remotely. I mean, there are one or two issues at the moment which mean I can't be with you, but I greatly appreciate the opportunity to give this talk. And as was said, this is about using mobile phones for low-cost disease diagnostics. And I'll just share my screen now. So as I said, and there are aspects of imaging in this, and there are aspects of using mobile phone, but actually the talk is underpinned by very low-cost paper technologies. And these are technologies which over the last, I suppose, 18 months or so, we have all become very familiar with. So on the left-hand side here, we have what is known as a lateral flow test. And these were developed actually by Unilever, or a spin-out of Unilever called Unipath in the 1980s for pregnancy testing, and are immunodiagnostic tests that look at antibody or antigen reactions on paper, and they have these very simple result markers, which are actually made by either silica or gold nanoparticles being accumulated on a test strip as a consequence of the binding reaction. These are very low cost, of course. And we all know from COVID that there are some difficulties associated with both the sensitivity and indeed the specificity of these tests. And the gold standard test, of course, is one which we refer to as PCR, polymerase chain reaction. And this has the advantage that it measures the DNA, which is present within the sample, and it has an amplification step in it. So there's an enzymatic reaction which takes a small amount of DNA and amplifies that so that you have a much larger amount of DNA before you do the detection step. So the clear advantage of that is that if you've got very small amounts of DNA, which might be present either in asymptomatic or pre-symptomatic people, then as a consequence of that, you can amplify that amount of DNA. You have an extremely sensitive test. The disadvantage, of course, is that these are primarily done within laboratories. And so they're done in centralized testing facilities where often you have to send your test away. So from the point of view of community sensing, having a lateral flow test is a great advantage where an individual or an untrained individual can read the test, but having these so-called PCR type tests where we're amplifying DNA has disadvantages in so much as we need quite detailed instrumentation. So our aim actually in this talk, and what I'm going to talk, certainly the first half of the talk about, is how we're going to put these very sensitive nucleic acid tests, DNA tests, onto paper strips, basically getting the advantage of both the ability to do community testing, but also the sensitivity and the specificity of a test in able to detect very, very low amounts of infection within communities. So actually, if we look at paper tests and the use of paper in diagnostics, we can see that there's a long history of this. I've already mentioned actually that Unilever were the first people to develop this for the pregnancy test, and they went on to license it to just about every diagnostics company in the world. Kodak, which was a photographic company in USA, actually used to make dry reagent strips before their demise. And there are a number of academics who've largely worked on immunodiagnostic testing, looking for biomarkers for disease and I would highlight George Whiteside's, in particular, Paul Yeager in Washington, Patrick Tablin in Paris, and Helen Lee in Cambridge, have all been pioneers of doing immunodiagnostics. But what we want to do actually is to not do immunodiagnostics on our paper strip, but to do DNA diagnostics, and indeed, as I'll show in a moment, RNA diagnostics as well. And this is actually a child that we're testing in Uganda here, and we are taking a blood sample and looking in this case for malaria. And in fact, we've worked not only in Uganda, but as you'll see during the talk, we've worked broadly in Tanzania, Rwanda, and also Malawi as well. And over the last, I suppose, 5 years, 6 years or so, we've developed these tests incrementally. And just to take you on this journey, we started out actually doing our paper tests in a reference laboratory in London, the London Malarial Reference Laboratory. We've moved on and worked in zoonotic infections and looked at the transmission of diseases between animals and humans. We've developed the techniques so that they can actually be used quite effectively within underserved communities with low resource healthcare settings. And then most recently, the two most recent developments have been moving the whole platform onto a smartphone, which I will talk about in the talk here today. And then most recently, actually moving away from malaria and looking at viruses. And one example that we're just in the process of publishing, this is in proof at the moment, is with an RNA virus called hepatitis C. But of course, COVID itself is an RNA virus, and I'll talk about the work that we've done on SARS-CoV-2 as well. So the concept that we have here is that we want to be able to work within communities where people are going about their everyday life, to be able to do diagnosis within those remote villages where there are perhaps no power, no running water, using a mobile platform to be able to provide decision support. So in what turn out to be quite complex multiplex tests, to be able to provide a support tool which is able to help an untrained person who might be a healthcare worker or perhaps a nurse to interpret that data, and then to be able to provide treatment in situ. at the actual place. So these are very typical of the testing environments which we have to work in, and you can see that there are very basic sanitary facilities, and where those facilities do exist, they're not necessarily terribly effective. Here you can see a toilet that enters actually into Lake Victoria, into where the village children play, and I will say something about that later. This is a region called Apache in central Uganda, which is in fact the most malarial place in the world, and it's a village surrounded by swamps, and as a consequence of this year-round wetland, there are enormous numbers of mosquitoes, and hence malaria is prevalent. And as I've said before, the communities which we're working in are ones which have very little in the way of resources, so no running water, boreholes for water, and no power other than perhaps solar or diesel generation. Now, the work, we often talk about interdisciplinary or multidisciplinary work. I'd like to think actually that the work that we're doing here is transdisciplinary, because not only does it cut between, for example, engineering, molecular biology, fluid mechanics, and perhaps public health, but also it's had quite a strong impact locally into healthcare policy, and in particular has enabled governors, administrators, and other healthcare authorities to redesign some of the treatment regimes which they're putting in place in these villages. So the logistics of carrying out this work is quite tricky. You need to have access to four-wheel drive vehicles to go into these remote communities. We generally take teams of students and postdocs into here. There's usually a group of about 6 or 8 of us who work together at a time. We get informed consent from the parents to test children, for example, and we actually also do a GPS logging so that we're able to do longitudinal studies on the families. And then working in schools primarily, and the school is a really good hub for the community where, for example, people can gather together. We take a variety of different samples for different diseases. So you can see a blood finger prick being taken from this young girl here, but we also actually take fecal and urine samples, and these are the children actually going out with little jars to take those samples. And we do a whole series of reference testing as well. And then the aim is that we're able to develop these diagnostics which enable us to do treatment in situ, within the community. So this is actually a picture of children being treated for disease schistosomiasis, which I will talk about in the second half of my talk. And they're being treated actually in the school, having been diagnosed. So moving DNA sensors onto paper actually is quite a complicated process, and this runs through the process. So in the case of malaria here, we take a spot of blood. We need to do some sample processing to remove malarial parasite from the red blood cell, and to lyse open or break open the parasite so we have access to the DNA. The DNA needs to be concentrated before it's amplified, and then resulting in a signal, and then that signal is read by our mobile phone and used within a decision support tool to give advice on what the infection outcome actually is. So the translation of these three major processes onto paper has been this challenge, which has taken us 5 years or so to do, going from that finger prick of blood to the visual cue, which we can see on the actual paper. And the key to doing this actually is embodied in this left-hand side image here. So here you can see our paper lateral flow cartridge, and interfaced with that is a second paper test. And we use this concept of folding the paper, so-called paper origami, in order to valve the reagents and the sample. So we put a sample of blood onto a piece of paper, and then if we want to move that to the next part of the paper, we simply fold that over and allow that movement to occur by capillarity occurring vertically between the defined channels in the paper. Now, what stops the sample from moving laterally is that we use hot wax printing in order to be able to define the features, to enable the sample to move through a series of processing steps before it reaches the microfluidic chip. And then actually on the microfluidic chip, we do the amplification process where we incorporate labels into the amplicons, the DNA that we're trying to detect, in order to give us our lines. So why are we interested in DNA sensing? Well, DNA sensing, as I've said, is very accurate. So it can be used for monitoring changes in prevalence of a disease outbreak. And of course, we've very much seen that during SARS-CoV-2. Unlike immunodiagnostics, it is actually quantitative. So when you do a lateral flow test, actually you're not measuring the amount of material which is present. And this is part of the reason why they're not so sensitive, is that the quantification is less good. Whereas actually for a DNA test, we're able to quantify the changes in patient-to-patient variability. I've already mentioned the fact that these tests are extremely sensitive, and there is the ability to look at either pre-symptomatic individuals, who have not yet shown symptoms but are carrying the disease, and also asymptomatic individuals, and those are ones who will be carrying the disease quite often in high loads, but actually are not showing any symptoms. And as we've seen actually in the recent pandemic, these pre-symptomatic and asymptomatic testings are crucial in being able to control the disease. And then the most important thing perhaps is the specificity, so informing personalized treatments. So if we look at the policies now of, for example, the World Health Organization, WHO, what they're very keen to do is to move away from mass community testing, which has really underpinned some of the major problems that we have in, for example, antimicrobial resistance, and move towards personalized treatments, wherein within a whole community, we can identify those individuals who are infected, and rather than treating the whole community for a disease in what's called a mass drug administration process, we actually inform ourselves as to which of the individuals are actually infected, how much disease they have within them, and consequently give the correct dosing accordingly. And actually, this is a quote from the World Health Organization saying what healthcare workers need are low-cost rapid diagnostic tests that require no refrigeration, no lab equipment or specialized training, that can detect parasites in low abundances. So if we look at malaria as our first case study, and it's the one actually that we've done the most work on, you can see the great success that there has been in fighting malaria. So on the left-hand side here, we see the prevalence of malaria in sub-Saharan Africa. And we can see that over a 10-year period, about 650 million cases were averted by using insecticide-coated bed netting and mass drug administration. And that was a great success story of fighting a disease. However, sorry, However, since about 2015, things have changed quite dramatically, and they've changed for a number of reasons. There has been the introduction actually of drug resistance into the malarial parasite, but there's also been other problems with the diagnostic testing, such that we now see that malaria is back on the rise again and is becoming a problem. And in fact, many of you will have seen just recently in the last 3 months or so work on vaccines, which one hopes will start to reverse this trend yet again. So this area of drug resistance is one which is extremely important. It actually emerged initially in Cambodia, on the border with Thailand, and has moved now into sub-Saharan Africa, such that the routine treatments that we have for these diseases are no longer as effective as they used to be. And this need to be able to inform our diagnostics and to tailor our treatments is becoming ever more important as a consequence. So here are some results of our lateral flow testing, and that you can see that the paper origami strip actually has been torn away here, and we've taken some very nice photographs. You can see clearly in the bottom here that we have a microfluidic arrangement, and in fact the amplicon arrives within this little chamber here of our paper microfluidic device. We have a blister pack which has got a running buffer in it, which when you squeeze with your fingers very simply, it enables the sample to move onto the paper. And this is what we call a multiplex test, where we're doing a number of tests at the same time. And you can see here that we've got actually only 3 lines working in these tests. So we've got our control experiment showing that the test has worked in lane 1, and then in lane 2 we have all of our different species of malarial parasites, so they're called Plasmodium pan, so there's all of the Plasmodium species. And then in lane 3 we have Falciparum here, which is the most prevalent species. And you can see a variety of different results which you can read, showing firstly that the test has worked, and we use a marker gene, which is a tumor suppressor protein, in order to, which we all have within us, to show that the test has actually worked successfully. And here we have different combinations where, for example, on the right-hand side, there are no Plasmodium malarial parasites present. And on the middle one here, we actually have no Falciparum, but we do have Plasmodium pan present. So we're able to get quite detailed information about the nature of the disease within the individual. Now, these were tests which were done within our laboratory, and you can see we get these very nice clear lines which are not difficult to interpret. However, when we go to communities and do these tests in communities, it all becomes a little bit more complicated. For a number of reasons, which include the cleanliness and of the testing environment and the extent to which competing forms of DNA are present locally within the environment. When we're doing lots of testing, there is naturally a lot of DNA, perhaps in the schoolrooms that we're testing in. We've got poor light conditions. We're never quite sure what the orientation of the device is. And the interpretation is very much more difficult. And so that led us to look at the mobile phone as a platform to help us with this. And as we know, the mobile phone has got a lot of sensors on it, and I'll say more about mobile phone sensing in the context of schistosomiasis in about 10 minutes time or so. So the sensors that we have, well, most importantly, we have a battery pack in there, and we use the battery pack in our testing, as I will explain in a moment. We have a GPS in there, so we can do some geolocation as well. We have a camera, which we use for recording the test. We have a communication system, which we use actually to send the test to the cloud, where we run a relatively simple decision support tool that does the interpretation and sends back the results from the test. Now, of course, smartphones are prevalent within societies everywhere, and in fact, within sub-Saharan Africa, we see some of the greatest increases in smartphone use that there are, and those are growing from quite a low base, but annual growth on smartphone usage now, for example, in Uganda, is one of the highest places in the world, grown by somewhere between 5 and 10% annually. So this is a tool which is becoming commonplace, and one which we're able to use within these communities. And this is the port that we use for our smartphone. In here you can see our plastic cartridge with the paper strips within it. What we've done actually is we've torn away our origami sample processing, and we insert the cartridge into this very simple circuit, very low cost, very simple circuit. This circuit actually provides the small amount of heating which we need in order to be able to do our DNA amplification process on that chip. So I spoke at the beginning about polymerase chain reaction, PCR, and the C in PCR is for cycling. And the key to how PCR works is that we're changing the temperature, cycling the temperature between roughly 60 degrees and 90 degrees. And in doing so, this cycle of enzymatic reactions is amplifying the DNA which is present. Now, this instrumentally is quite a complicated assay to perform. And instead of using one of these PCR reactions, we use a different amplification reaction, which is called an isothermal reaction. It works at one temperature, so we only actually have to heat up our chip to about 65 degrees, and then the amplification process continues until we stop heating. Very simple circuit. We've got a little power supply unit. We're using the battery from the mobile phone. We've got a microcontroller unit, and the microcontroller unit is primarily controlling two sensors, one of which is a reference sensor, and one of well, it's controlling the heater, sorry, and the feedback from two sensors, one of which is a reference sensor, and one of which is measuring the temperature on our chip. So simple circuit to build, and you can see that we can maintain very stable temperature on our chip for about, well, for as long as the battery lasts, actually, but we're able to run these for somewhere around about 4 or 5 hours, should we want to, and perform that DNA amplification within the device. So as we've got the mobile phone, which we're using for the battery pack, we also have an app in there, which is used to actually, as the human interface. We're able to enter quite a lot of information in there on where the device was actually manufactured, what reagents are being used, and those are all actually embedded within a QR code, which is on the test. We can enter anonymized information on the patient, so the patient ID, the time, the temperature, and so on, and any metadata that we also need to include in there. And as I said, we have this interface to the cloud, so we brought together a number of different tools in order to be able to do this. And so we have our databases, which are managed on a Google tool. We have some cloud tools. This API REST is used for the communication between our app and the cloud. And we perform this very simple artificial intelligence process within the cloud. So here you see that actually the results of our training, and the way that we did our training was to take a whole series of model tests where we had deliberately put errors into the test. And those errors might include, for example, false positives, false negatives. They might include very, very small amounts of DNA being present. They may have included faulty reagents. And then in the context of whilst we're reading the actual result using the camera, we put a variety of different lighting conditions, shadows, shades, orientations, so that we have this very large training set that then enabled us to look at the efficacy of this as a decision support tool. And you can see the confusion matrix here between our ground truth here and our actual measurement, and you can see that we do extremely well in getting very good decision support on our devices. And in fact, we use two different networks. We use ResNet and CNN. Both performed equally well. In fact, probably ResNet has some advantages in it, in so much as it has marginally lower power demands, and power is always at a precedent. One of the key aspects of the work that we've done on this actually has been the security that we have put around the device as well. And in particular, we've used blockchain for end-to-end encryption. And blockchain within healthcare, I think, has a number of great advantages. The fact that you can have a key to it, and actually that key enables only people who are trusted to access that data, is a really strong aspect to what we've been able to do. So, for example, we might lodge the key with ourselves, with people within a local healthcare facility, and perhaps within database providers within centralized Ministry of Health systems, who can access our data when they need to, but otherwise it is largely secured. And here you see some very nice proof of concept data that we had, just comparing some of the different techniques that can be used for malarial diagnosis. So the standard technique that could be used and was used broadly was using a so-called GIMSA stain with light microscopy, where you're looking for the parasites within the blood sample. And you can see there that 15 out of 100 times there is an incorrect diagnosis done. And that incorrect diagnosis is done despite the fact that trained healthcare workers are using the microscopes. Then we have our lateral flow immunodiagnostic tests, and you can see here that roughly 18 in 100 cases are misdiagnosed within those lateral flow immunodiagnostic tests used for malaria testing. And then here you have our decision support enabled paper DNA device, where we're almost exactly an order of magnitude better than the immunodiagnostic test. So we're missing 1.8 in 100 as opposed to 18 in 100 positive tests. So I think that's a considerable success in that respect. Now I'm going to move reasonably quickly now. We have done a lot of work detecting malaria, and the signature that we're trying to detect from malaria, where the genetic information is encoded within the parasite, is DNA. Now, there are a variety of viruses that exist where the information is encoded within RNA. And the RNA molecule itself cannot be amplified. And in order to amplify it, you first have to convert it back into DNA and then do the amplification as before on your device. So doing tests for RNA viruses is more complex. And as I said at the beginning, we've just done a very nice proof of principle study on this, which will be published in the coming weeks in Nature Communications, on a virus called hepatitis C. But one of the viruses which has been occupying our thoughts greatly over the last 18 months has been SARS-CoV-2, which is an RNA virus, where you need to do this back conversion of the RNA into DNA before you can do the testing. And that adds an additional level of complexity to the actual test which we're trying to do. So this is the work that we set out to do. This is our prototype device here that's got our lateral flow strip on the side here. This is a swab which is taken out of a mouth sample, a nasopharyngeal swab. The swab goes into the device, and the device is then heated up, and there are a number of different ways in which we can heat up this device, and we end up with a multiplexed measurement. And in fact, we're working with a UK company who I'll say a little bit more about at the end, who are called Mologic, and they're the same company actually that was spun out of Unilever and Unipath back in the 1980s, who have reinvented themselves several times since. And in fact, we're interested here in looking at three different viruses. So we're looking at a virus associated with a common cold, RSV. We're also looking at flu, influenza, and we're measuring SARS-CoV-2. So we've got what we think of as being a triplex measurement. In fact, there are many more tests than we have to do than three, because there are different forms of flu and RSV which we have to account for. But we're doing a triplex test that is able to distinguish between influenza, RSV, and SARS-CoV-2. And we've been testing this within Malawi, actually. So we've had quite a complicated study here of doing serum sampling, of doing PCR testing, of doing our isothermal testing, and looking at infection levels. And actually, the study involved comparing healthcare workers and non-healthcare workers. And so we can see measurements around the prevalence. We've got a limit of detection around 60 copies or so. And interestingly, what we see is not only the high levels of infection that exist within these communities, many of whom are actually asymptomatic, but also actually that there's very little difference between healthcare workers and non-healthcare workers in the levels of infection. I'll come back to the SARS-CoV-2 at the end and say a little bit more about that. What I'd like to do now is to switch tack a little bit. We're staying on the broad subject of using artificial intelligence and imaging. We're moving to a different disease, actually, and this is a disease called schistosomiasis. And this is a liver fluke, a helminth worm that lives within our intestines. And like malaria, it is transmitted through a vector, and the vector, instead of being a mosquito, is actually a snail, and the snail lives in fresh water. So actually, the liver fluke, or the worm, lives within our intestines, within the hepatic portal vein. The male and the female live together permanently in copula, producing eggs. Those eggs, depending upon the species, are either transmitted through the urine or the feces. When they go into fresh water, the eggs hatch, they infect a snail. The snail produces large, very large numbers of these parasites, which then actually burrow back through the skin of individuals either playing in the water if they're children, or working in the water if they're fishermen, or washing their clothes or gathering food, and reinfects the individual. And in Africa, there's about 250 million people who are infected with this disease. And if we look at the disease amongst children, the disease causes anemia, it causes poor educational outcomes from missed days at school, stunted growth from malnutrition. Ultimately, the worms damage the liver and can lead to cirrhosis and chronic kidney failure as well. And actually, due to changes which are occurring within diet, particularly in the context of increased amount of fatty food and uptake of alcohol, as well as other diseases which affect the liver, like, for example, hepatitis C. With the prevalence of all of these increasing, schistosomiasis is now becoming a disease which kills people. So it's no longer a disease that has high morbidity. It's a disease which is now beginning to have quite high mortalities associated with it. Now, interestingly, although it is a disease of sub-Saharan Africa, it's prevalent right the way across Asia and South America as well. The species specific, for example, to Japan, which are called Schistosoma japonicum, for example. And there are about 1.5 billion people at risk to this disease, many of whom live in communities that don't have access to water. So there are three possible tests which are done at the moment. So we have our immunodiagnostic test using a lateral flow strip, which is commonly used, but actually has low sensitivity and is not very good. There are PCR type assays which are performed in laboratory tests, but they cannot work within communities for the reasons that we've described already. They need to be run in large laboratories. And then we have microscopy, so light microscopy using a stain called the Cato-Cat stain. And this is this green stain. And here you can see a fecal sample of human feces, and the stain has been used to identify this schistosoma egg in the in the sample. Now, some of these samples are actually in the urine for some species, but the most prevalent one within Uganda, Schistosoma mansoni, is actually present within the feces. And so teams of healthcare experts go in with microscopes to do egg counting, and they sit all day looking at fecal samples in order to identify those individuals who are infected and those individuals who need to be given drugs. Along with schistosoma, there are a whole range of other helminths which are present, and they're all actually treated the same way. There's hookworm and pinworm, which are two other variations, but the WHO, for information, just has somewhere around about 6 priority helminth species that individuals within these communities become infected with. And actually, by light microscopy, you can identify each of these if you're trained and if you have the time and perhaps rigor to be able to do this. So if we look back at this photo that I showed, this again is in our school classroom. This is in Mayugi on the banks of Lake Victoria in Uganda. You can see that there are 2 microscopes here, light microscopes, which have to be carried in large boxes, probably need light sources as well. And the individuals are highly trained, highly skilled individuals who have to perform the staining test. And then here we have one of our colleagues here who works within the Ministry of Health using a little device that we have developed, which is based upon smartphone again. We don't use the smartphone imaging anymore. We started off doing that, but we just use a simple objective lens. This is a 3D printed device, which we can make within the lab. It costs round about £100 to make, including all the optics and electronics that we use. It scans relatively fast, so we can scan a slide in about 20 minutes or so. And there is some very simple optics in there with a simple objective lens. We use a tube lens, which we use for demagnification to give us a larger field of view, and then we render the image down onto a simple CMOS 12 megapixel sensor that gives us high resolution images. We use a mobile phone in this photo where we've developed an app which enables us not only to control the scanning, but also to collect data. And we collect that data as tiled images. And again, we use our same blockchain type interface, which we go to a decision support tool, which we can either run actually on the mobile phone or we can run in the cloud as before. And one of the nice things about mobile phones now is that you can use them as LANs, as local area networks. And in fact, we can run multiple devices using one phone and collect information from multiple devices. So this actually is a great testament to the PhD student, Oliver Higgins, who did this work, because during lockdown, he was unable to go to Uganda for long periods of time. So actually, he used social media to get training data, which he collected either as images or as slides, which people sent him from basically across Africa, around the world, actually. And he ended up with a training set of 6,000 slides, and then was able to look at the efficacy of this as a test. Now, we used a very effective image recognition tool, which is made by Google, actually, called Efficient Det, which we incorporated into this. You can see that we've got sensitivities in most cases of round about greater than 95%. Ascaris, which is this one here, is a difficult helminth to actually detect, partly because it's much smaller and the egg is much smaller, and the egg actually has a lot of comparability to fecal matter that you see within the sample. But anyway, for our confusion matrix, you can see here along the top where we've misdiagnosed out of 1,000 samples. There's somewhere around about 20 or 30 samples which have been misdiagnosed. Our vertical axis here is perhaps a little bit more worrying to some extent, because these are ones where we think that there is no egg present, but there are actually eggs present. And again, about 30 or so out of the 1,000 that we're missing. Now, we've been very lucky actually to be able to most recently work with Merck, who are a drug company. And if you saw the picture earlier on where the school child is being treated, you will see that they were actually being treated by a drug that's made by Merck called praziquantel. And Merck are part of the Global Schistosoma Alliance, which is a organization set up to help eliminate the disease. And the WHO has a target for elimination of the disease within the next decade. And we've been lucky enough actually to win a very nice global prize with Merck around being able to further develop this instrument for use within communities.
10 minutes.
So, yeah, I'm just going to finish up now. Just a few words at the end. We've worked, as I said, extensively in Uganda, but also in Rwanda, Tanzania, and Malawi, particularly Malawi actually for the SARS-CoV-2 testing. And you can see that we're now working with the Ugandan Industrial Research Institute. All of our tools, all of this information is available on open source, open access. In fact, if you go to the publications that we've worked on this, all of the code is present there, and there are videos showing how you can make these devices yourself. And in fact, we work with University of Makerere and sort of outreach programs run by university students and MSc students in developing this as a technology. We do quite a lot of public engagement as well. And here you can see a mosquito, which is an origami mosquito. And you remember the paper folding is exactly how we do the test. And so this idea of origami, which brings together the concept of the mosquito, the disease, and the diagnosis, is one which has received quite a lot of attention within Uganda. And then, of course, we all know about lab on a chip technology, but this is chip in a lab technology here. And you can see that we've got this little chicken that's come in to interrupt our studies. During the weeks where we were working there, we noticed actually that our medical disposal unit bins were getting raided by the children. And in fact, the children were taking the latex gloves out of the disposal bags and using mosquito nets and the latex gloves to make footballs. So they say necessity is the mother of invention. I should say just a little bit at the end. I mentioned about the first work that was done back in the mid-1980s on paper lateral flow tests for pregnancy testing with Unipath. And that company went through various iterations. At one stage, it was owned by a large diagnostics company, Alere. It's now actually independent again and called Mologic. We work very closely with them. And in fact, Mologic have just been bought by the George Soros Open Foundation and the Bill and Melinda Gates Foundation. So it is now a wholly owned, not-for-profit, open social enterprise, shall we call it. And actually, there is another social enterprise called DHIS2, which is spun out of Oslo, University of Oslo, which is a digital database provider. And we're now working as a collaboration across the three of us in this sort of open access, open innovation activity. So a big thank you to all the people who have funded this: Bill and Melinda Gates Foundation, Clinton Foundation, our own GCRF program. Lots of this has been funded by UK government, by the European government, by MoLogic, by our own Scottish government, as well as our National Institute for Health Research as well. So a lot of people who have contributed to the funding of this, and also students. In fact, many of these people were PhD students who have gone through the process of graduating based upon the work that they've done in this, and are now postdocs. Some of them are still in the process of finishing their PhDs, particularly Ollie Higgins, who did the work on the microscopy, and Gosheng, who did the work primarily on our decision support tools. So a big thank you to the Ministry of Health, the Vector Control Division in Tanzania, to Kilimanjaro Clinical Research Institute, and in Malawi to the teams involved in our SARS-CoV-2 program there. Thank you very much. Thank you.
I found that absolutely fascinating, John. Yeah. So what we'll do now, we'll pass it over to the audience. Are there any questions out? Want to ask anybody? Okay, there's one over there. Can we go? This is the microphone.
Hello. First of all, thanks for your presentation. My question was related to one of your last slides when you have this confusion matrix that you had difficulties in exactly this one in the empty class. Do you know what went wrong there, or you're still investigating what the problem?
So which are you talking about, the horizontal line or the vertical line?
You're not on mic, John, yeah?
Hello? Can you hear me?
Yeah.
Are you talking about the vertical class, or vertical or the horizontal?
Well, both of them.
Okay, so I mean, what's happening in the horizontal class is that we're confusing different species, so we're not making the correct diagnosis. So we might be saying something is Ascaris where it's not Ascaris, and in the vertical class, what we're doing here is we're saying that we were unable to detect eggs, whereas the ground truth told us that there were eggs present. So the ground truth would be normal light microscopy with an expert doing this, and our inference prediction would be one based upon our detection software. Does that make sense?
Yeah. I mean, I thought you had already investigated why you had this problem, or you still.
Well, actually, we haven't yet. But I mean, if I go back to well, maybe this slide here, and I'll just share it again, just to show you the challenge of what we're trying to do. So I should say there's a group in UCLA who work with mobile diagnostics as well, OSCAM, and they've looked at this problem as well, but they've looked at it in urine, and so there's a species of schistosoma called Schistosoma hematobium, which is where the eggs are excreted in the urine. And of course, urine is very clean and there's no particulates in there, and it's very easy to do that egg counting diagnosis. Within a fecal sample, and when you bear in mind the diet that these children have within these community settings, which is largely vegetarian, largely grain-based, lots of pollen within the feces, and in addition to that, the because of gastrointestinal infections and so on, quite often the feces have different consistencies. Actually, this task of identifying eggs, and not only identifying eggs as a sort of object detection, but if I go right back, not right back, but to here, to be able to distinguish, in this case, between schistosomiasis here, which has this characteristic hookworm, which doesn't have a hook actually, and pinworm here, you know, to be able to do that species-specific detection, I think is great credit actually to the Google detection software, amongst other things.
All right, thank you very much. If you want, I can drop a line of an email if you want to see how to improve those results.
Yeah, no, I'd be absolutely. Do you know? I mean, as I said during the talk, we're very, very open to collaboration on this. All of the collaborations we have, whether they're with Merck or Mologic or whatever, are all open innovation, open source. We're very keen to engender collaboration on this.
Sweet, thank you.
Thank you very much. Any questions on the other side? Nanjing, is it? Can you, do you want to ask the question?
Yes, I do have a question. Imaging showing on the screen now.
Yes.
Yes, the image is showing on the screen now. Is it taken by the mobile or is it taken by other maps?
No, the images I'm showing on the screen now are images which are taken off the web, actually, as examples, just to show you. But if I go back to here, these are real images that we have taken here.
Taken by a suited device and the mobile.
Taken using our very simple optical setup, you know, an objective, as I said before, an objective lens, a tube lens, and a CMOS sensor, basically, arranged within, with LED illumination, arranged within a 3D printed scanning stage. And actually, we do the focus using a simple flexure of the so the focusing stage is based upon flexure of that stage, bringing the CMOS in or out of focus.
Okay.
But you mentioned that you didn't actually use this image for analysis. You actually just using this spectra or transmission or something. So the imaging you don't use, and the imaging you're taking is some information you not use or what?
No, so I mean, there's two sets of data that we have. So there's a training set of about 6,000 images, and then there is a testing set of about 1,000 images, which we've taken ourselves.
Okay, understood. Thank you.
Okay, very good. Thank you very much. And can I just finish by thanking the organisers for being patient with me and enabling me to do this talk by Teams. So thank you very much.
Okay, just very quickly, any other questions before we close off? No one? Jonathan, absolutely fantastic work. I think the fact you're working on the ground in Africa and so on, in Uganda, I think is the challenges that presents are actually enormous, and people who haven't done that won't appreciate that. So I think this is outstanding work, and I think it's going to have a real impact going forward. So on that basis, I think we'll finish unless any more questions. Thank you very much. We'll reconvene at 10:30 and continue the workshop from there. Thank you very much.
Thank you.